this beautiful cytoplasm. Again, this is the cytosol or cytoplasm of a healthy cell. And this is one of our nice, long, skinny, lots of folds on the inside mitochondria. So let's add some glucose to here and see what happens when we add glucose to a healthy mitochondria. In comes glucose and turns to pyruvate. And you do not have to remember this long term, but just hang with me. The pyruvate then has a very large arrow that delivers and changes that energy cell to acetyl-CoA. So it starts as glucose, becomes pyruvate, and then the next big step is acetyl-CoA. You don't have to remember all this. I just want you to hold on to that word acetyl-CoA because of what it means in the next few slides. Once acetyl-CoA is inside that mitochondria, this beautiful cellular process inside this Krebs cycle takes the fuel that's found in acetyl-CoA and it runs those electrons down an electron transport chain. It uses some oxygen to add phosphorylation, then adds a phosphate to it using oxygen, and you end up with almost 90% of the energy coming out the other side of that mitochondria from that glucose in the form of ATP. ATP is just a scientific way of saying here's some energy, but when we talk about mitochondria being the fuel source or the furnaces or the factories of energy, this is what's really happening. When you look at the biggest pathway for energy being made, it must go through that delicate waves in the mitochondria, and out comes ATP. If you look at some side turns, I like to call these the farts of energy that come out of a mitochondria. You can have some lactic acid come out when it doesn't even enter into the major part of the mitochondria. So glucose to pyruvate happens at the edge of that mitochondria, right on the inside fold, and then out comes some energy a little bit, and you'll make a little lactic acid with that. If you do get energy into the acetyl-CoA and through that Krebs cycle, you'll get some succinic acid, or succinyl-CoA will convert a little bit of energy out. It's kind of like a trap door that escapes before it gets all the way to the end of the energy burn. Now, don't click the channel away. This gets much better. Hang in there. You're not going to have a quiz on that, but it really does allow you to compare when you say, oh, that one works this way. If I took glucose and I added it to a cancer cell, and in this case, we have that really short, stubby mitochondria with not very many folds. In goes glucose, and it does become pyruvate, but it is the major source of where a cancer cell produces energy. That arrow coming out the side is a very big arrow, and 20% of all the energy coming out of a cancer cell is from that little spot there. In our healthy cells, this is I like to call it the fart of, it's an electrical kind of nuance of energy. It's not the major source of energy, but in our cancer cells, they clearly depend on using that first little transition of taking glucose to pyruvate as a place that they make energy. In those tiny little dots, I like to think of the breadcrumbs that become acetyl-CoA inside the mitochondria. This then passes electrons into the Krebs cycle, and out from that tiny little breadcrumbs, the succinyl-CoA that comes out of a cancer cell mitochondria is 75% of the energy. Now, just back up a second. In our healthy cell, this was 10%, with each of those tiny little arrows coming out the side of our mitochondria, barely giving us 10% of the energy. The glucose isn't a different molecule for these mitochondria in the cancer cells, but instead it is a wasteful or very abnormal use of this fuel. And we're going to drive that home a little bit more. The other part where our healthy mitochondria put out 90% of the energy, in our cancer cell, a few of those electrons carry energy down to the electron transport chain. They do have a few of them that get phosphorylated, and about 5% of our energy from that little bitty fuel that made it all the way to the end of the mitochondria in a cancer cell is used. All right, hang in there. So here we have that when we look at how sugar is used in a cancer cell, it loves sugar and it really doesn't spin it through that mitochondria, as opposed to our healthy cells that do like sugar, like glucose, and when they use glucose, they get a very efficient production of energy. Cancer cells are known for being wasteful, while healthy cells are efficient. Let me explain what that means. So here's our beautiful healthy cell again. Again, that mitochondria is in the cytosol. It's a nice smooth cytosol, and the mitochondria is beautiful long with our wavy folds on the inside. That's a healthy mitochondria. Our glucose comes on in, goes into pyruvate. This really big arrow gives you a lot of acetyl-CoA. That energy gets really spun through that Krebs cycle and along the electron transport chain into the place where we have oxidative phosphorylation. And again, 90% of our energy comes out the other end. Only a tiny amount comes out these little bitty places where it isn't a very efficient use of energy. The most efficient use of energy is to run it through those mitochondria, push it all the way down the electron transport chain, and not waste a morsel of fuel as you produce energy for your body. Okay, so now let's show you again what happens with a cancer cell. So here's our little stubby mitochondria in our yucky looking cytoplasm. Our glucose comes into cancer cells. It takes it from glucose into pyruvate, and a huge amount of glucose is turned into lactic acid. I like to point out to young physicians, young students in medicine, that one of the old tests for blood tests to look if somebody had cancer was to measure their lactic acid. And we can study that in their body, but in a late stage of cancer, we can actually measure that lactic acid would build up in their body. Their body had become more acidic than it should. And the source of it was this wasteful place that glucose just quickly turned into pyruvate, and a ton of energy was made out of that glucose into lactic acid. And it was energy for the cells. These cancer cells take a lot of energy, mainly because they waste so much of it. Lactic acid was an old school test for, does the patient have end-stage cancer? Here's those little breadcrumbs giving a few drops into the acetyl-CoA, spinning those energy units through the Krebs cycle, and again, out that side chain coming a lot of energy from succinic acid using succinyl-CoA as a conversion. So as I look at the tiny little electrons that run down the side, they do hop into the end of the electron transport chain. You do get them phosphorylated, and only a little burp of energy gets all the way through the efficient part of that mitochondria. Again, cancer cells, mitochondria, as well as many other health issues in medicine, they have broken energy use. So as I look at just a quick comparison, I want you to notice lactic acid was 20%, succinic acid was 75%, and the extra energy is only that little bit. When you compare that to our healthy cells, 90% of their glucose, of their energy comes out. So the same amount of glucose can go in, but as you process that flow of energy, it changes dramatically when you've got a healthy, functioning mitochondria versus the mitochondria found in cancer cells. And this plays a major role in how we teach patients what they can do using the ketogenic diet for cancer. Okay, so here is the question where I would like you to click on and answer this question before I tell you the answer. And that is, do you think cancer cells like oxygen or don't like oxygen? We know our healthy cells like oxygen, but do cancer cells, does it make their world better or does it make it worse? All right, giving you just a moment to answer. Okay, so that's giving you a little time to answer that question, and I will show you that, yes, cancer cells do not like oxygen, and of course our cells do. Let's get a little bit more into that explanation. So here's the setting of your glucose coming in. We're going to now add the thought that oxygen is abundant. So once again, your glucose becomes pyruvate. A nice big arrow gives you a bunch of acetyl-CoA. The Krebs cycle spins those electrons down, and you end up at the major efficient delivery of energy through the electron transport chain. I want you to take a deep breath in through your nose, out through your mouth. You just pulled in oxygen and breathed out carbon dioxide and water. Your cells do that too, especially when they're using an efficient delivery of oxygen. You add oxygen to those cells, and in comes the oxygen, out comes carbon dioxide and water. We call this cellular respiration, and it is a sign of health and the efficient use of oxygen and energy in your mitochondria. Let's move to a place where oxygen is not as abundant. These cells are holding their breath. Actually, there just isn't enough oxygen for the energy demands around. And thankfully, there's a backup plan. Their glucose comes into pyruvate, and without oxygen, they are able to make lactic acid. They call this substrative level phosphorylation, but it's often termed fermentation. You'll see some of those cells go into acetyl-CoA, spin through the Krebs cycle, and without oxygen, they do take that trap door and become succinic acid while producing a little bit of energy through the substrative phosphorylation otherwise known as fermentation. So this is the backup plan your system has in the event that there's not enough oxygen around for the energy your system needs. Again, healthy cells can survive with no oxygen by using the trapdoors that were overly used in those cancer cells. All right, let's look at that cancer cell. Again, in the setting of no oxygen, what happens? Glucose comes in, pyruvate comes out, a nice big arrow for lactic acid becomes fermented. Yes, a few little crumbs turn into acetyl-CoA, the Krebs cycle spins out, and that trapdoor leaves you most of the energy coming out through succinic acid. The fermentation of glucose through a cancer cell in their broken mitochondria is most of their energy. That happens with oxygen and without oxygen. But here's a little foreshadowing that when you run acetyl-CoA through that Krebs cycle, there is a sneaky byproduct that is very dangerous for that person. All right, let's take a closer look at when oxygen is present. Again, there's that mitochondria with a goofy look to it. We have now a bunch of oxygen present. Here's our glucose. Starting out as glucose, 2-pyruvate, there's your lactic acid. Spin a few of those little breadcrumbs through acetyl-CoA in the Krebs cycle of that mitochondria. And now you have the addition of oxygen, and a few of them run through that electron transport chain for 5% of the energy. But in the presence of that oxygen, now you can see the cell trying to respirate is only a tiny amount, if at all, in some cancer cells. The punchline I want you to remember is glucose is a fermentable fuel. So is glutamine through succinate. These fermentable fuels are very important to keep cancer cells fueled and alive. And they are the strategies that we use to help patients have less delivery of energy for their cancer cells and better delivery for their own healthy cells. All right, let's do a quick review. Here we have cancer cells that love sugar and healthy cells that like them. We have the energy in cancer cells, which is very wasteful, whereas our healthy cells use the full length of their mitochondria for an efficient energy production. Cancer cells hate oxygen while healthy cells love them. Now let's move to ketones. So here again, we have our beautiful cytoplasm, our long, wavy mitochondria. We're going to watch to see what happens when a ketone enters our mitochondria of a healthy cell and we make energy from a ketone. Instead of starting with glucose, we're starting with BHB. You might have heard this as BHB is sold on the market today as a supplement. Much like we've learned to swallow glucose for energy, we can now swallow BHB or ketones for energy. The 3 products that I have on the market today that offer this as a supplement include the Keto BHB, which I affectionately call ketones in a can. I then have ketones in a capsule, as well as ketones mixed with MCT, called Max Strength Keto Combo. As you watch, that BHB becomes acetoacetate. Acetyl-CoA is in strong supply. Acetyl-CoA is the place where our 2 fuels now share similar components as they make energy. Acetyl-CoA is the first place that our journey for glucose and our journey for ketones become one. Acetyl-CoA is worth remembering because of how unique that is as a fuel source and as a source of energy. Whether that fuel came from glucose or ketones, it's spun through the Krebs cycle, run down the electron transport chain, the oxygen comes in, it breathes out carbon dioxide and water, and once again, our mitochondria has an efficient energy source. All right, let's see what our cancer cell does with ketones. Okay, here's our ketones. There's our goofy looking mitochondria for our cancer cell. And we take BHB, turn it into acetyl-CoA. It spins through the Krebs cycle. Some of those electrons dance down that electron transport chain. Oxidative phosphorylation pushes out energy. Is this how a cancer cell makes energy? The answer is no. Your cancer cells do not have the ability to efficiently use acetyl-CoA to transport them into energy. At the very best, it can take acetyl-CoA and push a few points of energy out the side, But remember, glucose is a fermentable fuel. It is fermentation that your cancer cells are using to produce energy. Glutamine is a fermentable fuel, but ketones are not fermentable. They must go through the full electron transport chain, turning into energy at the oxidative phosphorylation phase. All right, last but not least, I gave you a little bit of foreshadowing for those electrons. We're going to talk about reactive oxygen species, or free radicals. We know that a lot of those in our cells promote cancer. However, if you end up with many of them inside your healthy cells, they are very damaging. So let's take a look at, once again, no oxygen and our healthy cell. There's some glucose to pyruvate, a nice big arrow gives you some acetyl-CoA, you spin it through the Krebs cycle, but without oxygen, you have your lactic acid and succinic acid, and you are fermenting all the energy that cell can make. That is fermentation, no oxygen present.